Hello, my name is Professor Hudson. Let's continue with our narrated PowerPoint lecture for Chapter 5. Let's continue talking about how do substances move across membranes in Section 5.2. Let's begin by looking at a video of how diffusion works.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Now that we've had a chance to look at diffusion in a liquid, let's look at some other methods by which molecules can move and how these methods can be used for moving molecules in and out of cells. Diffusion is considered a form of passive transport. In passive transport, molecules follow their concentration gradient, which means they move from high to low concentration. Diffusion is not the only way in which molecules can move in and out of cells. This slide lists several forms of transport. The transport mechanisms fall into two categories: passive transport and energy requiring transport. The plasma membrane is differentially or selectively permeable. This means the plasma membrane only lets certain things in and out of the cell. With passive transport, the three common methods seen are simple diffusion, facilitated diffusion, and osmosis. No ATP energy is required, and molecules in these processes move from high to low concentration. In energy requiring transport, those methods include active transport and bulk transport. In these situations, molecules move against the concentration gradient, which means they move from low to high. This requires carrier proteins or a vesicle and or energy in the form of ATP. Let's watch some animations that will highlight some of these mechanisms. On this slide, you have a summary of the transport methods across membranes that were discussed on the previous slide. This slide has definitions from your textbook for each one of these processes. Please make sure you know the two main categories of transport across membranes, along with each one of the types of transport and some descriptions for each type as listed on this slide. So you saw an animation on how diffusion works with respect to simple diffusion. Now let's watch an animation on facilitated diffusion. Facilitated diffusion, the molecules are moving from high to low concentration. However, in this case, a receptor protein or channel protein in the membrane assist in their movement.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
So one of the key things with facilitated diffusion is that you have to have a carrier protein or channel protein to assist the molecules in moving across the membrane. But they still are moving from high to low concentration without ATP energy. Let's look at an animation of how osmosis works. So remember, simple diffusion, facilitated diffusion, and osmosis are all examples of passive transport. When we use the term diffusion, we can be referring to any molecule moving from high to low concentration. However, when we talk about osmosis, osmosis is a special kind of diffusion where the molecule that we are focusing on is water moving across a semi-permeable membrane. Semi-permeable means selectively permeable or differentially permeable. In osmosis, water moves across a membrane typically only permeable to water. Let's watch this animation.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
That was osmosis. In this animation, they illustrated how water moves from high to low concentration across a semi-permeable membrane. Now let us review. Question 13. Osmosis is specifically about the movement of blank in and out of cells. Choose the best answer. Remember, you should be writing down the letter that corresponds to the correct answer for each question you see in these PowerPoint lectures, as you will need to input these letters in the lecture quiz in Blackboard. Okay, so this slide illustrates three primary mechanisms of passive transport. Simple diffusion, which is a process by which molecules diffuse directly through the phospholipid bilayer of cell membranes. Molecules that move across membranes by simple diffusion include water, oxygen, carbon dioxide, and lipid-soluble molecules such as alcohol and vitamins, A, D, and E. Now, facilitated diffusion is a type of transport allowing substances to cross membranes with the assistance of special transport proteins. Water-soluble molecules such as ions, amino acids, and sugars diffuse down their concentration gradients with the aid of channel and carrier transport proteins. Two types of proteins allow facilitated diffusion: carrier proteins and channel proteins. Aquaporins are a specific type of channel protein that allows water to pass through. We also talked about osmosis. Osmosis is the diffusion of water across a selectively permeable membrane. Selectively permeable is in response to gradients of concentration, pressure, or temperature. Water diffuses from a region of high concentration to one of low concentration. Dissolved substances such as solutes will reduce the concentration of free water molecules and hence the purity of water. This can affect osmosis. The concentration of a solute in water determines the solution's osmotic strength or its tendency to attract water across a membrane. The greater the solute concentration, the greater the osmotic strength. When we talk about the concentration of a solute affecting the way in which water moves in and out of a cell, we are referring to the concept of tonicity. Let's talk a little bit more about tonicity. The term tonicity refers to the strength of a solution, or put another way, how concentrated the solution is. Isotonic solutions have equal concentration of water and equal concentration of dissolved substances. A cell in an isotonic solution will show no net water movement across the membrane. What this means is the rate in which water moves in the cell will be the same as the rate in which water moves out across the membrane. A hypertonic solution is one with a greater solute concentration. A cell placed in a hypertonic solution will shrink as water molecules will leave the cell at a greater rate at which water molecules will enter the cell. In essence, the hypertonic solution is dehydrating the cell. And a hypotonic solution has a lower solute concentration. A cell placed in a hypotonic solution will swell as the water will enter at a greater rate than when the water exits the cell. Osmosis is the movement of water through a selectively permeable membrane by simple diffusion or facilitated diffusion through aquaporins. Water moves across a selectively water-permeable membrane down its concentration gradient from the side of higher concentration of free water molecules to the side with a lower concentration of free water molecules.